membrane transport, and this time we'll be discussing facilitated diffusion, which comes under passive transport or downhill transport, and we'll be concentrating on this protein channel mediated facilitated diffusion. Now, channel forms close open pores, which means channels can be opened or closed as per the conditions. And we will be discussing aquaporins, which form exclusively water transport channels, as well as ion channels which will be controlled by ligands, which are called ligand-gated channels, or those ion channels which are controlled by voltage, which are called voltage-gated channels. First of all, we go to aquaporins. Now, aquaporins, they are integral transmembrane proteins, transport proteins, which exclusively transport water. They are definitely not ion channels, and were discussed quite recently only in 90s. This transport is actually based on osmotic gradient also, which means water molecules will be high in concentration outside the cell and will be taken inside by osmotic gradient also. This osmotic gradient definitely plays a role in its transport through aquaporins. Now, aquaporins are especially abundant in RBC and in cells such as those secrete fluids, for instance like tears, saliva, as well as kidney cells, which cause reabsorption of water. In all these cells or these tissues, aquaporins are distributed in plenty, and what happens is that water permeability will be poor when there is no aquaporin expression also. Now, transport of water by simple diffusion. It's a very slow process. Water actually gets through the hydrophobic core by simple diffusion, but it's a very slow process. But through aquaporins, it's very fast, as high as 1 million molecules per second is being transported. And structurally, aquaporins form 4-part clusters or tetramers, as you can see here. They are tetramers in cell membranes, which are embedded in cell membrane, with each of the each of the 4 monomers acting as separate water channels. As you can see, this acts as a single separate water channel. And each of these aquaporin monomers are composed of a bundle of 6 integral transmembrane α-helices. So, this is how it forms 4 separate monomers which form a tetramer, and each of them carry water molecules inside. The amino and carboxyl ends face the inside of the cell. Now, when we look more closely at the structure of an aquaporin monomer, you can see that there are hydrophilic amino acids which lines the cavity of the channel so as to interact with the incoming water molecules. And at the same time, there are hydrophobic amino acids also, which are placed outside which will interact with the hydrophobic core of the membrane. And there are some positively charged residues, amino acid residues, which are strategically placed along the path of the channel, which will actually repel the cationic molecules which want to enter into the cell. So it will repel cationic molecules as well as protons, H plus ions, that may get into the cell, and cause it may actually cause imbalance in ionic concentration. So this avoids the disruption of normal H plus ion or cationic distribution across the cell membrane. Even hydronium ions are prevented from entering the cell. Now we move on to ion channels. Now, ion channels are extremely rapid in their action. 1 million ions can be transported per second, and these are mainly involved in transport of cations like sodium cation or potassium cation, or anions like chloride anion. And ion channels are widely seen or widely operate during transmission of nerve impulses or transmission of electrical impulses. And they are highly selective for the ions they allow to pass through them. Now, ion channels may be ligand-gated or voltage-gated. First of all, let us see ligand-gated facilitated diffusion. And the best example to illustrate ligand-gated ion channels are this action of acetylcholine ligand So this is acetylcholine, here it's the ligand, and the ligand action on the ligand-gated receptor on a postsynaptic membrane. So obviously acetylcholine neurotransmitter is the ligand here, and the receptor, the acetylcholine induced receptors are here, which are the gated channels, and which remains closed now. Now, when an action potential arrives, what happens is that the synaptic vesicle actually releases the neurotransmitter in this synaptic cleft, and this is by a process called exocytosis, which we will deal in one of the future lectures. Now, as the neurotransmitter is released here, they will induce the gated channel to open. So this is what happens during ligand-gated opening of these channels or control of these channels. And as these gated channels open, what happens is that these sodium cations, they get into the cell like this. So this is what happens during ligand-gated ion channel operation. Now, this generates allow the action potential to continue, and this the channel remain open for a millisecond only, and acetylcholine will be rapidly degraded by acetylcholinesterase. And these are the steps involved. We can go through it, and we move on to voltage-gated ion channels. Now, in voltage-gated ion channels, what happens is that change in membrane potential, that is voltage difference, is what switches the ion channels to open or close. Whereas in ligand-gated, it is the release of ligand which induced a channel to open or close. Here it is a voltage difference which induces a channel to open or close. And voltage-gated sodium and potassium channels are most commonly seen or occur in nerve cells and is involved in transmission of nerve impulses. So the best example to illustrate voltage-gated channel is the sodium-potassium channel, which are present in nerve cells. Like we can see here, during resting potential of around minus 70 millivolts, what happens is that both these channels remain closed. That is, sodium channel is closed, potassium channel, both of them are closed at a voltage of 70 millivolts. Well, let us see what happens at around minus 45 to minus 50, as well as during somewhere around positive 30, plus 30 millivolts. So, at around minus 45 to minus 50, what happens is that the voltage-gated sodium channels are opened like this, and sodium gets into the cell like this. And here at this time, the potassium channel remain closed. And at around plus 30 millivolts, what happens is that the sodium channels are closed and the voltage-gated potassium channels are opened like this. So this is closed, and at this voltage, potassium channels are induced and they are opened. And potassium, actually, the potassium cation goes out of the cell like here. So this is what happens. So this is how the different voltages control these voltage-gated channels.